We are doing our presentation on methicillin-resistant Staphylococcus aureus, or more commonly known as MRSA, and I am Lauren Norwood. So this is introduction to MRSA. MRSA is one of the most commonly known bacteria that is resistant to methicillin, the antibiotic. It is extremely prevalent due to its resistance and is extremely hard to treat. One of the ways that you can catch the bacteria is by using used razors, dirty towels, or if you share cosmetics with someone who has the bacteria. That's why it is very important to clean your products and disinfect any surfaces that might be contaminated. There's also another way you can catch MRSA is by your pets. If your pets are infected with MRSA, most people don't realize it, but if you have an open wound or you have a cut, you can catch the bacteria. Hospitals are also known to transmit the bacteria due to MRSA being a nosocomial infection. It is resistant to UV lights and antibiotics, so it does travel around hospitals. Patients who have open wounds or open surgeries can catch this bacteria. That's why it is very encouraged to try and keep everything clean, sterilized, use your UV lights to try and treat for the bacteria. The methicillin resistance was first identified in 1961 by British scientists. It had already been, resistant to penicillin, which was one of the most prevalent antibiotics at the time, but due to overusage, the bacteria became resistant. So when methicillin was used to start treating it, patients would use it too much and too often, and the bacteria eventually developed a resistance to it. And this is one of the most challenging parts about treating MRSA.
Hi, I'm Lauren McKinney, and I'm going to discuss the characteristics and mechanism of the superbug that is MRSA. Resistance to methicillin drugs is caused by the MRSA gene, which codes for a penicillin-binding protein. This gene has a significantly less affinity for the beta-lactam drugs, which is the group of antibiotics that methicillin is a part of. Penicillin is a derivative of methicillin, and some of the other derivatives are amoxicillin, oxacillin, flucoxacillin, and dicloxacillin. Because of the lack of affinity for the beta-lactam antibiotics, resistance is gained by the Staphylococcus aureus. Because the beta-lactam antibiotics normally would prevent the synthesis of the cell wall of the bacteria, but because there's not much affinity for it, it's able to keep growing even in the presence of the beta-lactam antibiotics. I've got a picture that has a more detailed description of the mechanism of the MRSA gene that goes on to bind for the penicillin binding protein, and where it attaches to the peptidoglycan cell wall, causing the resistance.
Hi, this is Mandila. I'm going to cover the symptoms part of MRSA. Since MRSA generally started with the swollen skin and red painful bumps. It looks like just a pimple or spider bites, and when you touch that, it feels warm like fever. And if you press that bumps, it's full of pus and other kind of drainage. So if you remove that if you have to remove that pus, this MRSA pus, these bumps quickly goes into the body. If that's happened, you need the surgical drainage for that. Beyond that, sometimes some bacteria are confined to your skin. They are not removing from the surgical drainage. So if that happens, they goes into deeper and deeper, and they penetrate into the body, and they harm to your major part of the body, like heart, lungs, valve, and sometimes they can confine into your joints, wounds. So if that's happened, you need the surgical removal. The ultimate treatment or removing things is the surgery.
I'm Keely Hale. and I'm going to be talking to you about the diagnosis of MRSA. There's two main ways of diagnosing this disease. The commonly used is by cultures. They take a sample either from the infectious spot or the nasal secretions, because Staphylococcus aureus loves to hang out in your nose. So they usually take some nasal swabs of that, and they do a lawn onto the plate. The major plates used are tryptic soy agar and Euler-Hinton agar. And then antibiotic discs are placed onto the agar to test for the resistance. It takes about 48 hours to grow, and how you read this test is you look at the zone of clearance that is caused by the antibiotic, you measure that, and then reference a chart to see whether it is resistant or susceptible to the antibiotic. The up-and-coming diagnosis is by DNA. One of our main technologies right now that is commonly used is PCR, so where you replicate a certain fragment of the bacterial DNA that identifies that bacteria, and you sequence that. Once you have your sequence, you can put it into BLAST, which is Basic Local Alignment Search Tool. It has a full database of published genomes of millions of different species. So you just put in your fragment that you sequenced, and it tests it against the whole database, and it pops up the ones that are more similar to your sequence. And then there's also immunoassays searching for Staphylococcus aureus or MRSA. It usually tests for protein A, which is a surface protein that is on the Staphylococcus aureus bacteria. It is a faster diagnosis, but it is a lot more expensive.
Hi, my name is Bridget, and I'm going to talk to you about the treatment and prevention options. So, as Lauren mentioned earlier, that MRSA is resistant to both methicillin and penicillin. Our first line of defense is vancomycin. And if the bacteria has already penetrated the tissues, vancomycin doesn't do a great job. So the second alternative to that would be daptomycin. And in cases where vancomycin isn't good enough, outside of the US they use teicoplanin, but it's not approved here. Some alternatives that are used as combination therapies are ceftaroline, linezolid, quinupristin, and daptopristin. And they're also combined with high dosages of daptomycin and vancomycin together, which can clear the bacterial infection. The most important thing for preventing MRSA is to wash your hands. Wash your hands for at least 30 seconds in between patients, and also anytime you switch out your gloves. And in a hospital setting, it's also very important to thoroughly clean all your equipment, such as all your instruments that you use, and to disinfect, and also use UV